স্টার্ট করে দিন স্যার।
ওকে, মিউটটা কোথায়, ওটা তো দেখছি না রে। Okay, good evening, all of you. Hello. Hello. Can you hear me? Hello.
অডিও ঠিক আছে স্যার?
অডিও ঠিক আছে, ওকে ওকে ওকে, কিন্তু কিছু একটা প্রবলেম হচ্ছে বুঝলেন। Okay. Okay, good evening, all of you. You are welcome to the live session of aspects of biochemical engineering, NPTEL course on aspects of biochemical engineering. So I hope you are enjoying the course, and main purpose of this course is to highlight the application that how we can apply the biological sciences for the real, in our real field, real life. So you know that I initially of my initial phase of my lecture I discusses that how the engineering has been discipline has been generated, and most of the engineering discipline has been generated from the basic science. So now today I am glad that I have some lot of questions from the participants, and some of the questions that has been raised in the forum that I am going to discuss initially today. And finally, I request the participants to write their question in the chat box so that I can take those questions and try to answer your question. So first question that I'm going to discuss, that how that this has been asked by one student. I don't know his name, but some email ID is there. The question was how amount of inoculum effect microbial growth phase, specifically lag phase. Now, as you know that inoculum plays very important role in case of any kind of microbial cultivation process. And we know that we have 4 different phases. We have lag phase, we have log phase, we have stationary phase, we have a decline phase. Now, this inoculum should be first. This inoculum should be active. Am I right? And if it is not active, active means it should be in the log phase. When we take the inoculum, it should be in the log phase. So that when we inoculate, the lag phase of this, that should be as minimum as possible. So if you see in my PPT, I have pointed out here that inoculum is a small amount of material containing microorganisms that is used to start the fermentation process. And in bacterial physiology, lag phase is defined as the phase which is necessary for acclimatizing the cell to new environment. That we know, because you know that initially cells, because like I told in my presentation that we Indian, we stay in a tropical country, and when you go to the Western country, it is a cold country, and we take little time to adjust with their environment. Because the same thing happens to the microorganism when they go to the new environment. It takes a little time, which is called lag phase. Now, during this phase, the bacteria grow, but the size increases. But the population density, what you call the concentration is remain almost constant. The inoculum size recommended to vary 5 to 10% of the media to decrease the lag phase. So you know that this is very important. Not at 5 to 10%, then again related with the concentration of the living cells present in the culture. And as I mentioned before in my lecture also, when you prepare the inoculum should be in between mid log phase to late log phase. But and volume of the media should be 5 to 10%, and concentration of the cells is approximately 10 to the power 7 or so, like this. And it varies from the fermentation to fermentation. I hope this answer is this question is very good, that how much inoculum effect of the microbial phase, that inoculum volume plays very important role. If we increase more and more, it doesn't affect the system much. In addition to that, it reduces the working volume, so efficiency of the system will be reduced. So we have to optimize the volume of the inoculum that is added to the fermentation broth. Second question that is raised one the same student: how is it differed on inoculum development in lab culture and industrial culture? Very good question. Lab culture, when you work with a lab culture, it is a very small volume. We can do it very easily, and we can maintain the aseptic condition. Of one essential thing of the fermentation process is the sterility of the whole system. So that sterility we maintain very easily in the lab scale. Now question comes, what's happened in the industrial scale? So that we use the live stream, and that helps for maintaining the sterility in the different sampling port and other the inoculum transfer line and all this things. We try to do that. Now, when the question now, since the inoculum volume is 5 to 10%, I work with a fermentation industry where the citric acid is produced from the cane molasses. Now we have 200 cubic meter for production fermenter, and another is 225 cubic meter production. Now I told you just now that volume of the inoculum is 5 to 10%. Now 5 to 10% of 200 cubic meter is how much? It is 10 to 20 cubic meter. 10 to 20, 10 cubic meter means how much? 10,000 liters. Now this 10,000 liters inoculum is very difficult to prepare in the lab. So you have to prepare in the fermentation plant itself. So what you have to do, you have to prepare the seed culture in the lab, and this seed culture, usually we have a seed tank that has the capacity of 10 liters, and we through the research and development we find out that what should be the concentration of the seed tank of the cells, live cell in the seed tank, that we take to the fermentation plant, and then aseptically we transfer to the inoculum vessel and the growth media. So, so that is how we, then in the inoculum vessel your culture is developed, and then we transfer to the production fermenter in between the mid log phase to late log phase. Now question that arises, that how you quantify the microorganism. Now, in case of, we know that there are two types of microorganisms. One is called unicellular cell, and there is the multicellular cells. In case of unicellular cells, number is proportional to mass. So you can easily count. As per example, I can give the example of Saccharomyces cerevisiae. I can give the example of any bacterial cells, mostly the bacterial cells that can be, you can count. Unicellular cell usually you can count, but In case of multicellular cells, as for example, I can give a typical example of as fungi, as for example, that Aspergillus niger, Penicillium chrysogenum. If you see under microscope, they are filamentous in nature. So you know that you cannot count their number because they grow in a mass. So in that case, in case of fungi, what you do, we produce kind of spores. And these spores, we count the number. And through R&D, through research and development, we find out what is should be the optimum concentration of the spores should be present in the seed culture. And this seed we aseptically we transfer to the inoculum vessel where in the media, growth media. When these spores grow in the growth media, then these spores will be converted to the vegetative cells. And these vegetative cells is used as a culture for the production fermenter. I hope you understand that. So what I have written here, that inoculum effect is a laboratory phenomena that is described as a significant increase in the minimal inhibitory concentration of an antibiotic when the number of the organism inoculated is increased. So that inoculum effect generally occurs with the beta-lactam antibiotics in relation to beta-lactamase producing bacteria. In the industry, the inoculum, that is the seed culture for the production fermenter, is developed in the inoculum vessel of the fermentation plant. So this is the so inoculum inoculum vessel capacity of the inoculum vessel usually should be 5 to 10% of the production fermenter, and that you have to prepared in the fermentation plant, not in the lab. But seed culture, basic seed culture, you have to prepare in the lab. So that is the thing I want to continue. Now another student, he has asked a very important question, that what I mean to say that an infertile the human do not reproduce. So you know that I told you that one of the important characteristics of the living system is the reproduction, that we all know. But he was telling that regarding the infertile system that we have, then question is how reproduction is the differential factor between the living and non-living cells, as we are developing the hybrid which are non-reproducing. That's a very nice question. But my answer to this question is that yes, some of the living cells they cannot reproduce. They are but if you go to the plant sector, you go to the animal cells system, we have a lot of infertile system that we have. But that is not the only characteristics of the living system. Living system has other characteristics also. What are the other characteristics? One important characteristics of the living system is that they are very sensitive to the environmental conditions. As for example, I told you during my regular class that if so we are all human beings. Suppose I am walking here in a room, and all of a sudden the temperature of the room increases to 45 degrees centigrade, but we can run away from this room because we cannot tolerate, because our system is so sensitive, we cannot tolerate. But we can work at 45 degrees centigrade, provided if we increase the temperature slowly, so that your system is becoming acclimatized with that temperature at 45 after so many years, after a period of time, and then you can work at the 45 degrees centigrade. So the living system has two important characteristics. One is they are very sensitive to the environment, and there is they have the property of the acclimatization. Another important factor that I want to share with you, that you are talking about infertile system. I talk in other way. Infertile system, that living system, I told you that a characteristic of the living system is that that reproduction. One, if in addition to reproduction, they are very sensitive to environment. They have the acclimatization property. Every living system should discharge some kind of waste product. So these are the different characteristics of the. So one characteristic is absent, other characteristics should be present in the system. That is, I want to conclude that. Another very interesting thing is that suppose I want to talk about the sterility. Suppose I have two bottles of milk. Suppose we sterilize the milk that and then we in a seal bottle we sterilize the milk and keep it for maybe for 2 months, 3 months, 4 months. Will you get any kind of change? What is your answer? Because you killed all the living system, nothing is there. But still, you can expect some kind of changes because that when you kill the microorganisms, you are not killing you are not denaturing all the enzymes. Some enzymes may present inside the cells, and these enzymes can participate in the reaction and change the composition of the milk. That is also there. That kind of thing that we have. So here, the question that has been asked, that the answer of this question is like this: living system has several characteristics besides reproduction. So in case of infertile system, the other characteristics of the living system are to be taken into considerations. As for example, different enzymatic reactions, acclimatizations, sensitive to the environmental condition, etc. I hope you understand this. Second question, next question that is asked, that is the vitamin. The vitamin C, whether it kills, what does this mean? Because vitamin C as such doesn't kill any bacteria or viruses. Because vitamin C doesn't kill any bacteria or viruses, but it is needed for the growth and repair of the tissue in all parts of your body. That in the now in case of COVID, pandemic situation, vitamin C also find very important role because doctors they prescribe that all the persons should have some kind of vitamin C intake so that this infection can be reduced to some extent. So it cannot kill, but it is needed for the growth and repair of the tissue of the all phase, and it is used to form the important protein used to make skin, tendons, and ligaments and blood vessels, heal wounds, and form scarred tissues. So this. I hope it is clear that vitamin C is nothing but ascorbic acid, and ascorbic acid plays very important role for our for the growth and maintenance of our body, of the living cells. Now, another student has asked some technical question, that is the B is calculated on the basis of thermodynamic efficiency on the anaerobic process as 0.7. So I hope the question is asked that why it is 0.7. So I told you that the purpose of this stoichiometry of the bioprocesses is three different purposes. I told you. One is it gives the intermolecular relationships, and second, it gives you the information that validity of your experimental data, that we can do that. Another we can find out that the amount of heat generation during the fermentation process, particularly aerobic fermentation process. Now we know, as you know, that most of the fermentation process usually takes place close to the ambient temperature. That is the basic difference between the biochemical process and the chemical process. Chemical process usually take place as a high temperature and a high pressure, but biochemical process usually take place as a low temperature, is close to the ambient temperature. But in addition to that, biochemical reactions are mostly exothermic reactions. Exothermic reaction means the temperature increasing. So we are in India, and India we have different climatic conditions throughout the year. We have winter season, we have summer season. In summer, the temperature increases, rises as high as 40-45 degrees centigrade, and winter the temperature goes down as low as 20-15 degrees centigrade. So in case of winter, that temperature goes down, so we require some additional heating system. The problem arises in case of summer, because summer the temperature rises more than 40-45 degrees centigrade. So ambient temperature is more. So your suppose your fermentation temperature is 30-33 degrees centigrade. Naturally, you have to have some kind of cooling arrangement. So this is why that this calculation is amount of heat generation calculation is very much required to find out to develop the proper environmental condition for the growth of the microorganism. Now, now the question that is asked, that thermodynamic efficiency. Thermodynamics efficiency is nothing but equal to the eta plus epsilon. What is eta? Eta is the energetic yield coefficient, plus and another is the product yield coefficient. Am I right? So eta plus epsilon. Now in this, so in case of this problem, this question was asked on the basis of this problem, that this is on the kind of anaerobic digestion process, the elemental analysis of the refuse that contain about 76% weight by weight of organic matter of the composition of this, and remaining 24% is as minimum methane production of the anaerobic is to be calculated per kg of the refuse, and also determine the stoichiometry of the equation. I told you, whenever you write the stoichiometry of any kind of bioprocess or biological system, you have to have some kind of idea on this process. Because we know in the anaerobic digestion process, the organic matter is degraded to, finally it will be degraded to the methane and carbon dioxide, also some kind of ammonia by the deamination reactions, and some organic acid might be present in the medium. So that, so we can write the equation like this, that what is shown here, that how we can generalize write the equation for the anaerobic digestion process, that C is 2.10 0.9 in 0.15 gives B moles of this methane, C moles of carbon dioxide, D moles of ammonia, and moles of hydrogen ion concentration. Okay. Now question comes that B, that we assume, this is in case of calculating the thermodynamic efficiency. For calculating B value, we assume the thermodynamic efficiency of the anaerobic digestion process is 7, because this is the basis through which we can find out the validity of the experimental data. In case of aerobic process, this thermodynamic efficiency varies from 0.5 to 0.6. And in case of anaerobic digestion process, it is 0.7. This assumption you have to make. This is assumed. This is the through research and development, they have found that this is the assumption you have to make. For the anaerobic process, this is 0.7, and aerobic process is 0.5 to 0.6. And on the basis of that, we find out the validity of the experiment. That I discussed during our presentation. I hope you understand that. The next question that is asked, does the product yield is somehow dependent on biomass? The question is yes, and the answer is yes, because obviously the product yield between the microbial fermentation process, the biomass plays very important role. Am I right? So Now we have seen there are two type of products are there. One is called three type of products. In fact, one is growth associated product, another non-growth associated product, and there is the mixed growth associated product. Now, what do you mean by growth associated product? Growth associated product means, here it is written there, you can see here, that is dp by dt is proportional to dx by dt. What does it mean? Rate of product formation is directly proportional rate of growth of the cells. And if it is this, we call it that growth associated product. Now, in case of non-growth associated product, and this happens, this growth associated product example is the ethanol fermentation process. It's a single cell production process. What is single cell protein? Single cell protein is nothing but microbial protein. So that here, there, the rate of single cell protein formation, rate of ethanol formation is directly proportional with the rate of cell mass formation. So this is the rate, it is the rate factor. But in case of non-growth associated product, as for example, citric acid, as for example, penicillin, as for example, streptomycin, the rate of product formation is proportional to X. A, what is X? X is the cell mass concentration. B is the non-growth associated coefficient. Am I right? So that also depends on concentration, but that is the concentration of the cell mass. So in case of growth associated product formation, rate of product formation depends on the rate of cell mass formation. But in case of non-growth associated product, which is usually take place at the stationary phase, and that is proportional to the cell mass concentration. I hope you understand that. The next question is that how we can calculate the degree of reduction of the ion. Very important, very interesting question. But the answer to this question is that, that what is degree of reduction? Degree of reduction, I told you, number of free electrons. Am I right? That is required for particular atoms. So, as for example, that carbon has the degree of reduction is 4 because it can take 4 electrons. Am I right? Or it can share 4 electrons. But oxygen, it can always take 2 electrons. Hydrogen can donate 1 electron. Nitrogen can donate 3 electrons. Like this, we have that. So degree of reduction represent what I have written here, represent the electron per unit carbon of the substrate, biomass, and product respectively relative to valence state of carbon each. Each compound involved the electrochemical reactions has a degree of reduction based on the electron donating and receiving capacity. As for example, oxygen, what if oxygen it cannot donate the electron, because if you look at the outermost orbit of the oxygen, it 6 electrons are present. So 6 electron, it cannot go out from the outermost orbit. What he can do, he can easily take 2 electrons, and so that just to make the orbit complete. 8 electron present in the outermost orbit, then it will be stable. So naturally, that oxygen can accept. So what is the degree of reduction of oxygen is -2. But in case of hydrogen, it can donate 1 electron. Am I right? Donate electron means it is liberating. So this is +1. So like this, we can calculate an atom is oxidized if its oxidation number increases. So oxidized means it is liberating electron, and reducing means it is accepting electron. So that is the things that we have. What I have written here, an atom is oxidized if the oxidation number increases. The reducing agent and the atom reduce if the oxidation number decreases. That is what I want to submit. Another question has been asked, that how we can multiply e to the power minus k1 into t. Now k1 is the rate constant. Am I right? T is the time of reaction. K1 is the rate constant and T is the time of reaction. Am I right? So, that we can have some kind of value, that is plus or minus value. So I have in the bottom I have written here, you can see, if the value is e to the power 1, this is plus 1, then that is equal to 2.71. Because, and if e value is minus 1, it is 0.3678. I think any scientific calculator, you have e value. So you can easily calculate the e value. So it is very simple. Again, I am repeating that K1 is a rate constant. Am I right? So rate constant is the time of reaction. If you multiply, then you will get a number. And number may be positive or negative. My number may be positive or negative. If you e to the power positive, then you have you can in the calculator you can just type, you will get the value of that. And negative also, I have given the examples here. E to the power 1 is equal to 2.71, and e to the power minus 1 is 0.3678. So I can write e to the power minus 1 1 by e to the power 1. Am I right? So 1 by 2.71 is equal to 0.3678. I hope this is very simple. You can do it very easily. Now, next thing that that question that is asked, that is that the Km conversion factor. That what is this mentioned that this conversion is wrong. I hope that you just check. What I want to point out here, that suppose Km value is 5 nanomoles. What is mean? Nano means 10 to the power minus 9. Am I right? So I can write 10 to the power 5 into 10 to the power minus 9 gram mole per liter. Is it correct? Now, at the same time I can write that 1 cubic meter is equal to 1000 liters. 1 cubic meter is equal to 1000 liters. So if you want to convert into the gram mole per cubic meter, then what will be then you have to multiply by 10 to the power 3, because then what will be there? 5 into 10 to the power minus 6 gram mole per cubic meter. I hope you understand that this is a very simple mathematics. You can do it very easily. I hope it is not a problem for you. You can do it very easily. Next is that one assignment 5. The question has been asked that can you explain how to solve the sum? That KT value is given here. KT value is given here. So how you can calculate the KT value? This is suppose the KT, the T half is the half life. T half equal to how you can write T half? T half equal to ln2 by kd. Am I right? So the kd is the if you divide by kd value, you can find out T half equal to 77 hours. This is a very simple calculations. If you just put your pen to it, you can get the answer. So I hope you get half-life period. The expression is ln2 by kd, and then that ln2 is equal to 0.693, and kd value if you put it, and then this T half value you can, and this is very much required, particularly in case of enzymatic reaction, because stability of the enzyme usually determined with the help of the half-life of the enzyme. If the half-life of the enzyme, because we know the proteins are costly, and at the same time we know that all proteins are not enzyme. So naturally enzymes are more costly than protein. So the major problem is that that major issue that we have, that is how to increase the stability of the enzyme so that we can use the enzyme for a longer period of time. So that is the major issue, and that is why T hub value plays very important role. So T hub value can be determined like this. That is ln2 by kd. So this is about the question, and some assignment. I have some numerical that calculation. We have some silly mistake. I hope I have given the answer in the forum. I hope you get the answer of that. Now I shall go to the forum and try to uh, try to share your question and try to answer that your question. Okay. Do you have any question? Hello?
No, sir. No questions in YouTube.
No question? Okay.
No, sir.
So what should I do? That should I continue that, or did I?
Yes, sir. You can close the session.
Okay. Okay. Thank you very much. Okay. Thank you. Thank you.